Hello. Can you hear me?
Yes, you're audible.
Okay. Mental health problems don't define who you are. They are something you experience. You walk in the rain and you feel the rain, but importantly, you are not the rain. Matt Haig. A very warm and wonderful morning to each and everyone present behind the screen today. I am Vinaya, second year BSc Psychology student of LISSAH College. On behalf of the entire psychology department, I welcome you all to the day 4 of the webinar series conducted in connection with World Mental Health Day. Before getting into the program, let me give you some general instructions. For keeping the quality of the sound functional during the presentation, please mute yourself. I repeat, please mute yourself. If you have any questions during the presentation, type them in the chat box and we'll bring them up at the end of the presentation. The session will be streaming live on our YouTube channel. The link will be provided in the chat box. We are looking forward for your cooperation and support in making this event a grand success. On this wonderful day, I feel privileged to extend my warm welcome to everyone. I would like to welcome the Management Committee of LISSAH and the guest of honor, Dr. Binu Ramachandran, to this webinar. Once again, a warm welcome to all the dignitaries and to all my lovely people out there. Today's topic is the new era of brain science, the molecular life of brain. To talk about this topic, we have a wonderful resource person with us. To introduce him, let me welcome Shanna Shamsuddin, second year BSc Psychology student of LISSAH College. Over to you, Shanna.
Good morning. Today, I'm here to welcome our guest of honor, Dr. Binu Ramachandran. I'm grateful to have an opportunity to welcome him on this special day. Dr. Binu R took his PhD in neurobiology from Leibniz Institute, Germany. He worked as an assistant professor in the Department of Zoology. He was a research scientist in Synaptic Signaling Groups, Germany, and also postdoctoral fellow at University of Zurich. He's also a reviewer of journals, Neuroscience Letters, Neuroscience, Neurochemistry, and so on. His honors and awards for his works are DST, SCRB, Terre Fellowship for Research Intelligence, DST, SCRB, ECRA Young Scientist Award, and Visiting Professor at Germany Center for Neurodegenerative Diseases, and Sharek Medical University, Germany. Thank you, sir. Thank you. Over to you, sir.
Thank you, Shamna. I'm audible to everyone. Hello.
Yes, sir, you're audible.
Okay, thank you. Thank you very much for this nice introduction. And also I thank all the management of the LISSAH College, and also especially to Dr. Emerson for inviting me for this talk. And yeah, and also the faculties of this department and also the institute and also dear students. I just want to share my experience in the field of neuroscience and how the brain works and the latest cutting edge technology in the field of neuroscience, and also some neurodegenerative diseases that are associated with the brain. And also it is very important keep your brain fresh, healthy, especially when the world they celebrate the or they announce the world mental health week of this month. And also I'm also thankful to all of you and all the participants, and thank you everyone. And let me share this slide, the presentation slide. Is it visible? Hello?
Yes, sir.
It's visible. Yeah. Okay. Thank you. Okay, let me start with the topic of today's talk, the new era of brain science, the molecular life of brain. So before we go into details, that we should really, all the neuroscience, all the techniques or everything starts with the unforgettable contribution of two eminent scientists of neuroscience, especially the Spanish neuroscientist Santiago Cajal, and also the Italian physiologist Camillo Golgi. And this Santiago and Santiago Cajal and Camillo Golgi, they were working almost all the time period. That means during the 18th century. And even this all the techniques, especially the optical techniques, are not fully developed, or even very good microscopes are not fully developed during that time. They used simple lenses to identify different ultra structure of the tissues. And from their observations, and these two pictures, these are simple drawings of the Cajal and Golgi from their observations of the nervous system. And just imagine and how beautiful these pictures are. And even today, with the ultra modern techniques or the imaging techniques like confocal imaging and the slit microscopy, and all these techniques even also provide the exact copy image of the same structure of the nervous system. And both of them, they won Nobel Prize for their ultra structural organization study of the brain, and they received the Nobel Prize in 1900 or 1906 in physiology and medicine. So all the neuroscience or the neuroscientists, all of them, they start with the pioneering work of this Cajal and Golgi. And this Golgi, we also know still people, they use the Golgi staining to identify the ultrastructure of the tissues and brain. Okay, so these are the basic science. These people are the basic pioneer or the pioneers. They basically contributed their entire life to the neuroscience. And then when we look at this brain, and you will see a number that we have more than 10 billions of neurons in the brain, and these are spatially and are restricted, and also they are oriented different regions of the brain, and they are interconnected and they integrate. And also the different informations are coded in different regions of the brain. And also during the retrieval, all these informations are retrieved, or they are they are integrated from the different sensory cortical areas. So, and when you go really go to the ultra structure of the brain, you will see the simple cells, that the neurons, and these neurons have more than 10 or more than 1,000 synapses. These are small junctions that basically connect the adjacent neurons for their effective communication. And these synapses, and then when you look at this structure of the synapse, and you will see this is the symbol, this is the symbol structure of a synapse, and you will see, and these synapses here, you will see this is a part of the synapse that usually we call the presynaptic terminal or the presynaptic region. Usually this will be an axon endings, but still there are different types of synapses that exist in the brain. Like this is usually when we say when we talk about the synapses, that usually we consider the axonal dendritic synapses. But you can see in brain different types of synapses, like axonic synapses are there, dendritic synapses are there, and then the dendrites are even they are connected to cell bodies, and also they are also on the synapse or the dendritic branches of the same neuron can even make synapse with the same neuron form autopsis. So there are different types of connections that exist in the brain for effective communications. And when you look at the general structure of the neuron or the synapse, you will see a presynaptic terminal here, and this presynaptic terminal is followed by a region of or a intermittent or a small spatial structure exists in between two adjacent neurons, and then you will see a postsynaptic neuron. Usually this will be a dendritic region. And when you look at this normal synaptic transmission and the impulse that reaches the presynaptic terminal here, and then you will see a lot of channels, especially the voltage dependent calcium channels are distributed this membrane of the presynapse. And then when this inaction potential reaches, and this basically reaches to a specific threshold that opens the voltage sensitive calcium channels that present in the presynaptic terminal. So as a result, a lot of calcium influx that happens into the presynaptic terminal. So that means a lot of calcium. This calcium is the main or the basic cation that trigger all the cellular metabolism. So everything, not only the synaptic transmission, all the pathways inside the cells, that starts with the calcium dynamics. So when this calcium enters into the presynaptic terminal, and these increase the level of the presynaptic calcium is sensed by different calcium sensors like synaptotagmins. So there are different around 17 isoforms of synaptotagmins are present, but they have different functions. Some are present only in the presynaptic terminal, and some of them are distributed postsynaptically, and some are distributed both pre and postsynaptic regions of the synapse. So when you look at this structure, the presynaptic terminal, then you will see a lot of calcium sensing proteins are present in the presynaptic terminal, especially the synaptotagmin 1, and then or synaptotagmin 1 and then synaptotagmin 3 on 4. Okay, not 3, the 4 is there, and this will sense the increase the level of the calcium inside the cell, and that triggers a lot of cascade inside the presynaptic terminal, like the synaptic vesicles are present here. And these synaptic vesicles that undergo priming, and also, or maybe it's a kind of activating the synapses for release, and you will see a lot of number of different types of synaptic pools. Synaptic vesicles are there. Some of them are readily releasable pools or reserved pools. So there are different types of synaptic vesicles are present in here, and then it's prime or it docking basically means it's interaction with the different molecules of the presynapse, and then after the docking, then that will goes to and then primed and then it released from on that. Usually that goes here in the presynaptic terminal, or that fuse with the presynaptic membrane and then it releases the synaptic vesicles or neurotransmitters from the presynaptic terminal. So this is the basic mechanism of release of the synaptic vesicles from the presynaptic terminal. There are different theories that also exist for the release of the presynaptic releasable pool of the neurotransmitters. For example, something like a kiss and run theory is there that basically explain this neurotrans or synaptic vesicle that fuse or kiss with the presynaptic membrane and then fuse and then release, and these vesicles membranes are recycled back and then it again refilled with the neurotransmitter. This is the way of the things or the processes that happens at the presynaptic terminal, and this is followed by the release of neurotransmitters. So in addition to the neurotransmitters that are released from the presynaptic region, you will see some receptors are also attached to the presynaptic membranes, and these most of the presynaptically occupied receptors that act as the retrograde messengers. So they can also receive messages from the prosynaptic neuron, and that can modify the presynaptic terminal through these receptors. So, and then once these neurotransmitters are released into the postsynaptic neuron, and this will bind to the postsynaptic receptors that are present in the membrane or the synaptic region, and you will see these receptors are distributed in the different regions of the synapses. Something like this is the synaptic regions of the that means the region that is basically that face the synaptic cleft. And in addition to the synaptic receptors, you will see a lot of receptors that are distributed here in the extrasynaptically and also the perisynaptic regions of the perisynaptic receptors are also present. So synaptic, extrasynaptic, perisynaptic receptors are also present in the membrane. And in addition to these, these receptors that are present at the postsynaptic neuron, that basically organized in different manner, and also their mode of action is also different. For example, some of these receptors or postsynaptic receptors, they are simple ion channels. For example, then when the simple cations or whatever anti-anions, whatever it, or simple ions, that when it binds to the postsynaptic receptors, that will simply open up, and then it allows the entry of the cations or anions into the postsynaptic membrane. But some of these receptors, they are coupled, or they are simply, we can say they are G protein coupled receptors. So because once the neurotransmitters that binds to the postsynaptic membrane, and this will trigger a lot of cascade in the receptors, and that also affect, or these are basically the transmembrane proteins, and they have a cytoplasmic phase and a extracellular space here. And this when the agonist or a molecule or a neurotransmitter that binds to the postsynaptic receptors, this will trigger a lot of cascades in the postsynaptic membrane that basically trigger a lot of cascade or a lot of signal pathways inside the postsynaptic neurons. That needs a lot of modification. So this is the general mechanism of synaptic transmission that happens in between the neurons. But in addition to the pre and postsynaptic regions, for example, the presynaptic terminals and the postsynaptic terminal, these simple synapses, these are also covered with some other extra processes like a astrocytic process. Astrocytes are there. So we also know about the glial cells. And these astrocytes, these basically guard or basically protect the synapse, and that also basically keep the homeostasis of the or the homeostatic plasticity of the synapse. So for example, and these astrocyte processes are present here and here, and once these neurotransmitters are released from the presynaptic terminal, and this one binds to the postsynaptic region or the postsynaptic receptors, and once these transmitters that binds to the postsynaptic receptors, there will be some conformational change that happens in the postsynaptic neuron, and as a result, the conformation of the synapse or of these receptors will change. And as a result, the affinity or the ligand binding affinity of neurotransmitters will be reduced, and these neurotransmitters will be released to the synaptic cleft. And if the neurotransmitter that exists in the synaptic cleft for a long time, that will further excite the cell, or that may also that also leads to excitotoxicity of the cell. So in order to prevent this kind of excitotoxicity or the imbalance of the homeostasis, these astrocytic processes that basically that helps to remove these neurotransmitters from the synaptic cleft, and they can process, and they can also recycle back, and they can also release different types of transporters of the neurotransmitters, and with the help of the transporters they can simply take or remove the neurotransmitters from the synaptic cleft. This is the basic mechanism of synaptic transmission. Okay. And then we comes to the neuronal synapses. This is a single image or a calcium imaging of a single neuron. And then you will see this is the cell body, and this one is the dendrite. All these processes are the dendrites. And you will see here there are small spine-like structures or some small protrusions are there. And these are, we call it as the dendritic spines. And these dendritic spines, here you see this dendritic spines that are, these are small spine-like structures that present in the dendritic membrane, and these regions are the dendritic spines are considered as the point of synaptic contact. So actually synapse or a axon from another neuron that makes synapse with this neuron through the dendritic spines. Okay, so there are different types of dendritic spines. I'm not going to details. And usually you will see a mushroom shaped dendritic spines are present. These are considered as the mature spines. And then these are these mushroom shaped structures here present. It has a head and a neck that is attached to the dendritic shaft, and then everything happens like the all transportation, all the signaling molecules, everything is run in between these mushroom spines and the cytoplasm of the neurons. And you will see basically in the neuronal transmission, you will see different types of neurotransmitters are present. But here, when you look at the basic neuronal transmission, you will see the principal cell or neurotransmitter of the brain is the glutamate or the excitatory neuron. And also you will see the inhibitory neurons are there here, that is also mediated by the gamma butyric acid, or be simply called as GABA. And these excitation or the glutamate transmitters and the GABA transmitters, these basically contribute normal synaptic transmission of the brain. So, and this glutamate that is released from the presynaptic region, and that binds to the postsynaptic neurons and it has different receptors like AMBA receptor, NMDA, and kainate receptors are there, and these are also work differently, or functionally they are different, like they are ion channels. For example, the kainate receptors and AMBA receptors are basically a simple ion receptors. For that means when the glutamate released into the presynaptic or the synaptic cleft, and this one basically activate the simple ion channels like AMBA and kainate receptors that basically allow the entry of sodium channels through the AMBA receptors and kainate receptors. But there are some other voltage dependent calcium channel, voltage dependent channels like a NMDA, N-methyl D-aspartate glutamate receptor is there, or simply we call it as NMDA receptor is there. And this NMDA, basically they are very sensitive, or no, not they are very, the opening and closing of the NMDA is very, that depend on the voltage of the postsynaptic membrane. For example, when the voltage, when the glutamate, first the glutamate is released from the presynaptic region, this first binds to these receptors like a AMBA, NMDA, and a kainate receptors. And but at the first, the AMBA will open, and the AMBA will open, and then it allows the entry of more sodium into the cell. And when it reaches to a threshold, and because usually the NMDA receptor of the postsynaptic neurons are clogged, or they will be blocked by magnesium ions, and these magnesium ions can be released only when the postsynaptic membrane reaches to a threshold, or to a specific voltage. So when the AMBA first opens, and then it allows the entry of more sodium ions into the postsynaptic membrane, and then more calcium and more sodium enters into the postsynaptic neuron. As a result, when it reaches to a threshold, and then the magnesium block of the NMDA will be released, and then it automatically open, and this NMDA receptor that allows the entry of both sodium and calcium channels. Okay, in addition to these, there are some other metabotropic glutamate receptors are there. They are just simply, they are G protein coupled receptors that basically allows the entry of calcium and sodium, or they simply we can say this, they allow the release of, or the entry of calcium ions into the postsynaptic membrane. And also that helps in the release. We also know this, the endoplasmic reticulum, or we simply called as neurolemma, neuroplasmic reticulum. And these the endoplasmic reticulum of the neuron, they act as the storage houses, or storage regions of calcium, internal storage of calcium channels, calcium ions, and that releases calcium into the postsynaptic neuron, and then that helps to increase the calcium level of the cell. So this is the way usually the glutamate works. The same way the GABA is an inhibitory neuron that basically allows the entry of the chloride channel. Then these cells become more hyperpolarized. So basically, when you study the basic nature, or the action potential, so we also know these action potential are small. They are very small, or we can simply say they are short or brief, refractory, all or known phenomena whose amplitude will not change with the time and space. This is the simple definition of the action potentials. And when it reaches, usually you also know this resting membrane potential and action potential and everything. So, and when the chloride channel opens, this cell polarity, or this voltage will become, or the cell become hyper polarized, or that means it's become more negative. And when the glutamate binds, this become or the cell potential become more positive in nature. So in this way, the normal synaptic transmission happens, and this also helps to or the balance of the glutamate and GABA that keep the neuronal momentum. And if anything happens in the normal transmission of the neurons, that will lead to different pathological conditions. Like sometimes if the imbalance or the balance is lost, the GABA will be dominated, or if GABA dominates, or okay, in that situation, then the cells become more depressed, and that leads to the depression stage. And also, or the glutamate, or if glutamate dominates, then the cell become excitotoxic, and you also know this become usually the seizure and then leads to epilepsy in nature. So any imbalance of these normal GABA glutamatergic synapses that leads to different pathological situation of the neurotransmitters. In addition to these neurotransmitters, there are other different types of neurotransmitters exist, like serotonin, then dopamine, and so all these different types of neurons that basically act as the modulators of the brain, and also they have specific function, or they also mediate specific transmission at specific regions of the brain. For example, in dopamine, that also helps in the that also involved in these dopaminergic neurons that present in the substantia nigra of the midbrain, and that also helps to relay different dopaminergic transmission to different regions of the brain. So this is the way usually the synaptic or the synapse works. And then when we go back, or when we come to the later stages of the synapse, though, so we know this, how the neuronal transmission happens. But when we go back to, and that we know this transmission happens through the active or the coordinated activity of the excitatory and inhibitory neurons. But how these memories, so because they are simply communicating each other, but we don't know how the memories are formed in our brain. And there are different theories. A lot of scientists, they have studied the process of the memory formation. And in early 1940s, the Canadian psychologist, Donald Hebb, through his observations, he also proposed a theory how the memories are formed. And according to him, the activity, the coordinated activity, for example, when an axon of cell is near enough to excite cell B, repeatedly or persistently takes part in firing it, some growth processes or metabolic change takes place in one or both cells, such that A's efficiency, that means the cell A's efficiency as one of the cells firing B is also increased. That means basically when an axon of cell A is near enough to excite cell B. For example, when a cell makes synapse with a dendrite of this cell B, and it repeatedly and persistently takes part in firing it. So that means basically when they interact with each other continuously, that will leads to some functional as well as some structural changes happens both cells. That means here they say some growth processes. The growth process simply means the structural changes of the synapse, and the metabolic changes that basically indicates the functional status of this synapse. That means the structural changes, that means basically the neurons will, or the synapse will develop new processes, or they have additional receptors in the membrane. As a result, these newly added receptors that helps to increase the voltage entry into the postsynaptic cell, thereby increase the efficiency of the cell B. So that means when the memories are formed as a result of the interaction or the reciprocal interaction of the adjacent cells or the interacting cells, that leads to the memory formation. So, and this is simply we call it as synaptic plasticity. That means the both the structural and functional changes of the pre and post synaptic cells that happens as a result of the continuous excite or continuous activity of both neurons. Okay, this is the Hebbian hypothesis or Hebbian plasticity, and this is still considered as the basic mechanism of memory formation at different level. Okay, and this theory postulates that the cells that fire together and wire together, that means the repeated or the intra-reciprocal interaction of the cells that leads to the memory formation. And we also know the memories are the physical representation of the memory, we simply call them as engrams, and these engrams are formed in different regions of this is not the memories are not clustered in a single region. We cannot simply pinpoint this is memory, this is. We can say that there are some modification that happens at the synaptic level, and that modification of the synapse are considered as the memory or physical representation of the memory or the engrams. And there are different theories that exist to explain how the memories are or the physical representation of the memory in the neuron, like the clustered plasticity model and diffused plasticity models. And the clustered plasticity model of the memory that basically explain the memories are formed as clusters of dendritic branches in different regions of the neuron. So that means these memories are exist as small modifications of the synapses as different clusters of the neuron. But some of them, they also propose that it is not like that. The neuron or the memories are distributed all around the neuron, or that means they are diffused throughout the neuron. At any point, they can the memories can be marked or represented as engram in a uniform manner. So, but still, both theory or both mechanism that exist in our brain. Okay, so that is the theory of memory formation or the Hebbian postulate, and this is followed by. So this was he proposed the idea of how these memories are formed or how the memories are formed in our brain. But later in 1970s, a group of scientists from, for example, Timothy Bliss, and this is the Timothy Bliss, and this is Sergio Lomo, and this is Professor Per Anderson. They started studying in vivo models, like in rabbits. They started studying how the memories are formed in our brain, and they were doing different stimulation in different, or they inject current into different regions of the brain. Especially they selected this hippocampus. This is the hippocampus region of the brain. And this hippocampus, this is a cross section of the brain, and you will see this is and this hippocampus has a different regions, like it has two types of cells exist in this hippocampus, like the CA region, and that DG is there, the dentate gyrus, and the CA1, that is the corno-amons region. This is the pyramidal cell body layer, and this is the granular cell body layer of the dentate gyrus of the hippocampus. And then again, this CA region is divided into this into CA1. Here there is a small region. So there is a small region that exists in between these two, and the CA2 region is here, then CA3, and the dentate gyrus. So the information of the brain that reaches to the dentate gyrus through this perforant pathway, and then it reaches the CA1, and then CA2, and then CA1. Sorry, CA3, CA2, CA1, and it goes back, and then the memories are formed, or the short-term memories are processed there. So this is the way of information happening in the brain. And they experimentally they induced different types of current, and they found there is an increased synaptic transmission when they give a small stimulation of the perforant path of the hippocampus. And they found this is and this, when after giving this stimulation, they found that there is an increased synaptic transmission. And this is they described as the long-term potentiation, or LTP. And they simply described this is a persistent increase in synaptic efficacy after a brief tetanic stimulation of the afferent. And this was the first evidence of the experimental or the existence of memory in the brain. And they can experimentally prove this. This is a cellular correlate of the memory formation that happens at the brain. And they also found there are different types of something happened. Hello. Hello.
Sorry, you're audible now.
But I don't know this. Okay, okay. It's stuck now, in between stuck. This is not moving actually. This slide is not shifting. Sorry, let me give a minute. Hello.
Yes, sir.
Okay, it's not moving. Let me
It's audible, sir.
audible, but it's okay. Okay. Okay, this was the contribution of Bliss and Lomo in the field of neuroscience. And they also found this LTP has different stages like our normal memory, like a short time phase and a long time phase. And they divided this LTP into an early and a late stage that based on their requirement for the protein synthesis. And later, okay, it's not still, it's not moving. Let me reopen this slide, okay?
Okay, sir.
Hello? Is it visible?
Yes, sir.
Is it visible?
Yes, sir, it is visible.
Okay. Okay, thank you. And they also they also found that there are different stages of the LTP, like our memory, an early phase and then late phase. So that basically depends on the how the frequency of the incoming inputs are stored, or whether the brain need that information to be stored, or so that depends on different processes. And also then it come that we also know the basic mechanism of memory formation, and these neurons that basically decide the information that or the incoming information, they are in the form of frequency coded, and then the strength of the frequency that determines the nature of the memory. And also then it consolidated, or during the memory consolidation unit, protein synthesis and other mechanisms. Okay, so this is the basic mechanism of memory formation, and this is followed by, and these are the simple system they used to study the electrophysiology from the brain, or especially these are the field recordings from that you can record signals from different regions, and also from different cells at the same time. So this is a simple neurophysiology rig used to measure the field recordings. That means that you can measure the response of a neuron, response of different neuron, or simply you can record from a field of neurons. Okay, and then this is the simple diagram that basically shows the how the record, or how the signals are recorded from the hippocampus. And this is the pyramidal neuron. You can record the or record the cell body response, and this cell body we call it as then population spike, because this is simple. When you record from a single neuron, that you call it as the action potential or a symbol as spike. But when you record it from a population of neuron, then you call it as PS, or population spike. And then usually we record it from the cell body, and also you can record it from the dendritic branches like the field DPSP, because this is also from a field. That means you will see a lot number of neurons around and number of branches around, and that's the reason it's called as field DPSP. Or simply you can or if you record from a single dendritic branch, we call it as excitatory postsynaptic current, and this is excitatory postsynaptic potential. That is from a group field DPSP. And here you see this is the recordings from the hippocampus slice. And this is usually used to record the response from a field of neuron. But later in 1970s, that there are different discoveries that leads to the recording of single ion channels from the neuron. This was by Professor Erwin Neher and Bert Sackman. And they discovered this method of identifying the single channel conductance from a neuron, and they awarded the Nobel Prize in physiology and medicine, and both of them from the Göttingen, Germany. And also you can record, see here, these are small pipettes that used to record the single channel conduction from a neuron. Okay, so this is the technology, the emerging technology that was of 1990s. And later, and this is the patch clump setup. You can basically record it. This is the patch clump setup used to record the single cell recording, and it also use a lot of electrical connections recordings, and used to measure the frequency of neurons. And this is the pattern, or you can simply study the firing pattern of the neuron. And then this is followed by, this was the technology that starts with because Bliss and Lomo, they started with the recording of the brain using they used actually small micro electrodes, and these micro electrodes are usually they are invasive in nature. That means when you go deep into the layers of the neuro or of the tissue, that basically pierce different cells, and also that cause a lot of damage. But later technology, and this was a simple pipette that these recordings are used to measure the ionic currents from single channel ions. But later the technology developed to how can we avoid this kind of invasive nature of recordings. For that one, in 2000, I think late 2000, the Professor Carl Dizot of the MIT, and they developed some ion or a specific ion channels like the channel rhodopsin. These are basically like our iodopsin of our retina. They are really sensitive to light, and these are isolated from primitive bacteria, and they found these channels, they are really sensitive to light. So they used these genes to activate, or they transfected, or they also used to, or they made transgenic line by incorporating these channel or channel membranes, incorporating genes, and they developed the different animal models, and they used some specific kind of light frequency, because they are just emitting, or they are just activating the channels to activate the ion channels to open. So usually we also use the electrical or the electrodes to basically activate the cell. So during that active, or when you place the electrode in the tissue, that will also make a lot of damages in the membrane of other cells. But so that was the drawback of these sharp electrodes. But when you express this kind of proteins or these channels in the membrane, so basically these channels are, or normally the channels that basically allows the entry of sodium and calcium ion channels, okay, that basically changes the membrane current, or the membrane potential. So instead of that one, they used this light, or the light sensitive channels expressed in neuronal membrane, and that are activated by some specific frequency of light, or that means the channel rhodopsin. Here you can see the channel rhodopsins are expressed here, and during light on, and these channels will open, and that also allow the entry of sodium and potassium on all these channels through these. So then the neuron starts firing, and when this lights are off, then it won't open. So this is the way of doing experiment, and this is not invasive. So these are the modern technology of optogenetics. Okay, and then this is followed by so this is the way usually they make the genetic construct encoding the photoreceptor, and they are expressed in animals, and then they manipulate or to find out or they stimulate the cells with a different frequency of light, and then they record the brain activity. Okay, this is the basic mechanism, and this is also followed by the latest technology of chemogenetics. Chemogenetics, they basically, in chemogenetics, this is also the latest technology of expressing ion channels in the membrane. In chemogenetics, what they use actually, they design some specific type of channels, and then they incorporate these specific channels into the membrane of or neuronal or any membrane, biological membrane, and then they use specific agonist of the channel and to open and to activate. Okay, so these are basically most modern technique that nowadays because these are simple chemicals, you can simply add the chemicals, and then these chemicals basically these chemicals bind to this, it's predetermined receptors, and then it opens, and then it allows the neuronal firing. So this is the latest technology of activating brain cells through chemically. Okay, and So, and then this is also followed by, at the same time, the era of artificial intelligence in life science and medicine. And we also know the artificial intelligence. This is also used. It's a special program that used to do a lot of research in biological science and of medicine, and they used to decode the brain, and also they collect the information, and you can also predict the release probability of the neurotransmitters, and you can play with these software specifically. You can it's just a kind of simulations, and you can play, and you can also change the concentration of calcium inside the cell, and then you can also predict the what happens at the synaptic level, and also you can basically the calcium dynamics of the cell, and also through you can use this technology, artificial intelligence, to detect the facial expression. For example, nowadays the people use this to study the autism. So in early stages of the development, for example, the age of the kids with less than 3 years of age, they basically recognize their facial expressions at the very early stage, and then and they can also predict, is there any possibility of getting neurodevelopmental disease in these kids. So this is the vast area of, and I think the artificial intelligence has a very vast application in medical and life science. So this is the latest technology reaches in neuroscience. Okay, and then we go back to our basic mental. These are that we were talking about the technology that reaches, or that drags everything to our friend until this moment. And now we go back to because this is more associated with our mental health and well-being, how we how the things, or how the neurons, or the health of the world population goes, or which direction. So we also know that this is still, the pandemic is going on. A lot of people get different mental stress. So a lot of things will happen. So we will get that the consequence of this the COVID in few years only, because the people will get a lot of mental stress, like even in kids also. So when come to the mental health of people, then this memory and forgetting, that because we have the memory, that is the ability to code for information, and also retain the information, that is we simply call it or the ability of the brain to retain information. Okay, that is we call it as, or the relevant information. We cannot record all the information that we get from the environment. Okay, some of them are significant, some are not significant. So the brain always tries to identify the relevant information of the environment, and then try to incorporate. And also this is happening, the consolidation happens during, and the same like the like a sleep also. So during sleep also, we also know the system, the brain will shut off all the sensory inputs, and then it try to process everything. Okay, so like that, the memory and forgetting, when we come to the forgetting mechanism, forgetting is also important for the brain, because when you learn each and everything that happens to the daily life, then that will be really difficult for the brain to sort out all the information. So the brain itself has a mechanism to clear out all these things, and what they will do, they will try to remove the unnecessary things or unnecessary memories, and then also keep it brain fresh. So this is the way usually. So so the memory and forgetting is very essential for our daily life. For example, and also we have proven this one, and we also identified a new mechanism of this that means the mechanism of forgetting, and also this forgetting is very essential for our human survival. And this was that we found that this protein, that is synaptotagmin 3, this is also involved in the forgetting of memories. And once you, and this helps in this synaptotagmin molecule that helps in the helps in the forgetting of unwanted or unnecessary memories, and then it keep it brain fresh. Okay, and this was also, this is in this work was we published in Science in 2019, and this we found that when you knock out or knock down the synaptic plasticity 3 in the brain, and this animal will develop, the animal will develop short-term memory. Even a small memory or a short-term memory can or the short-term memory will be converted into a long-term memory. So each and everything will be, the animal will remember each and everything. So at some point the animals reaches to a peak or reaches to a ceiling of or storing information. After some time the animal unable to learn further. So that means when you knock in, that means when you resubstitute the receptors, then it also regain the memory. So that means this molecule is very essential for forgetting. So that means basically the memory and forgetting are very important. Not only the memory, but forgetting is also important for our mental health. Okay. And then we come to different neurodegenerative diseases like this Parkinson disease, or you can say the poster disease, the gait disease, or that this is also due to the imbalance of the, or you also know the motor disease of the brain. This is by the dopamine or the neurodegeneration of the dopaminergic neurons of the substantia nigra. And you see, and also the people also exhibited different types of symptoms, some like mental issues, and they have also problems of walking or motor skill symptoms, also the tremors and everything exists. And these dopaminergic neurons of the midbrain nucleus, that means the substantia nigra, usually that degenerate. But the exact reason for this is not well known, but there are few studies that, some studies that also showed that even if pesticide, some kind of pesticide like a rotenone can also induce this Parkinson's in humans. So and these people found that this is usually used as a pesticide, the rotenone, and these pesticides also disrupt or that also degenerate the brain dopaminergic nucleus, leads to Parkinson disease. So we should be really aware of these pesticides and their usage and everything. So because we don't know exactly what happens, because there are a lot of neurodegenerative diseases are caused by these different environmental chemicals. Okay. And then this dopamine, usually when the dopaminergic neurons that release a lot of means normal dopamine is here, and that binds to postsynaptic. But in Parkinson's disease, this is also reduced the release of this dopamine, and also leads to the motor coordination, the lack of motor coordination. And this is usually the dopamine is synthesized from an amino acid tyrosine, and here is the synthetic pathway. And also this Parkinson is caused by different mechanisms like gene mutation, exposure to chemicals, and so there are different issues are there, or even our environment can also influence this kind of neurodegenerative, or the environment can act as an inducer of these neurodegenerative diseases. And that comes to the level of the schizophrenic patient. We also know that because this is the overexcitation, the dopaminergic theory that basically explains, because usually the dopamine has an excitatory two excitatory receptors are there, that is the D1 and D5 receptors are there, and these these are connected to this is a kind of G protein coupled receptors that basically activates different pathways and leads to gene transcription. But the overexcitation of this one leads to hyperactivation, and also different types of problems in like in hallucinations, and also depression. So there are different and anxiety, and so it also exhibit these are neurodegenerative diseases. And also these are also the etiology is also from our environment, and also prenatal exposure to hunger, and also neurochemical irregularities, and also some light exposure, so some chemicals, so viral infections. And so there are a lot of reasons that also contribute this schizophrenia in the population. And then it comes to the world of the Alzheimer's disease. The Alzheimer's, we also know this is a neurodegenerative disease, but there are different theory that also explains the origin of the Alzheimer's disease, like the cholinergic hypothesis and all the hypothesis of this, because the neurodegeneration of the acetyl or cholinergic neurons leads to Alzheimer's disease. And this was also identified by a British physician, Dr. Alois Alzheimer, and also and the disease now named after him. And this is an Alzheimer's theory. There are tau theory, the cholinergic hypothesis is there, then amyloid beta hypothesis is there, then tau hyperphosphorylation theory is there. So there are different types of theories that basically exist the development of Alzheimer's disease. Okay, so all these things, then here you see the cross section of the or the whole brain, and you see the its architecture, and here you see a lot of this brain is really shrinked, and these cavities are very high. And so this is the normal Alzheimer's patient's brain looks. And here that this also the symptoms are here, and this is from the also show a kind of social withdrawal, and then confusion, difficulties in completing familiar task, and so on, memory loss. So there are a lot of things, and even people also develop amnesia also, that we also know that we have different type of retrograde and anterograde amnesia is there. So the people will have the same problem. So we should be really careful doing means also we should also at the same when we protect the people, also we should also protect our environment to protect the mental health of the people, and also our physical as well as the mental health of our people, that because the environment can also influence the physical and mental health of our humans and also animals. And then that's it basically, and the mental health is very important like our physical health, and also that's it basically. And also I wish all of you keep cool, and also do exercises, and also keep your brain healthy, and do a lot of things. And these are the things that we should really should know what going on. And also, and especially in the case in this time, the pandemic of COVID, we should be aware of the things what's going on, and we should keep your brain still fresh, and also go forward. Thanks. That's it basically. Thanks a lot, and thank you very much. Hello.
Thank you, sir.
Okay, thank you.
Guys, if any one of you have any question to ask, please put it up in the comment box. Thank you.
No questions? Hello, am I audible?
Yes, sir, you're audible.
Okay, okay. I just went through the peripheral side of the what's going on this one, and you can also ask, feel free to ask what you have in your mind about the brain, because you people also study the psychology. Yeah, you will have definitely a lot of questions, and yeah, you, because this is actually a part of the experimental psychology, so of course you will have some kind of questions. Nothing. Okay, I got one related to the future of optogenetics. Optogenetics basically, future means it's already gone. This optogenetics is also gone actually, because it started very long back with the people. They started recording the brain sciences, brain recordings through the sharp electrode. Then these sharp electrodes are almost, not almost, or half, more than half of percentage is replaced by the glass pipettes, like patch clamping recordings, and then still that is also replaced by the optogenetics theory, or sorry, optogenetics tools. And then this is also, now it is all not completely replaced, it's not replaced completely, I'm saying the modification of this. Now it came, chemogenetics comes. Okay, optogenetics, the future of optogenetics, that means that basically for example, that I think maybe you can, you can apply a lot of hello. Okay, is there any? Okay, so you can apply, especially in the treatment phase, because even you also know, the in case of Parkinson disease, I think that was a long back that the people they introduced, because if there is no other choice to treat the Parkinson disease, the people they introduced stimulating the midbrain nucleus with the deep brain electrodes, like the deep brain stimulations. So that was very long back. But later, because in deep brain stimulation, they basically they implanted different electrodes in the brain, and then they have a small chip that is inserted or they're placed under this, or like a pacemaker, like a small chip, or a small device that used to on and off. And when you switch on this one, then that will basically uh, stimulate the midbrain nucleus and then release dopamine, and then that will coordinate the motor movement. This was the very long procedure, and that also I need to get a lot of ethical uh, um, clearance and everything. So this was but in optogenetics, that we need to, because you don't know exactly, because when you use sharp electrode, you will damage a lot of neurons or tissues, not only from the neuron, but you can also record it from different cells, like cardiac cells and muscle cells, etc. And when you use this kind of micro electrode, then you will damage the rest of the cells. Okay, but when you, and if you use, instead of this using this micro electrode, you can, when you express some kind of this, or you do optogenetics, you can basically you can stimulate, and then you can opens the channels. And so this is a kind, it's a kind of emerging technology, not emerging, it's already there, and they used in different pharmacological industries to find out different types of neuronal stimulation, and also to allow the neuronal activation. So this is the future basically. So they're used in different technologies of neuronal studies, and other they use the optogen. This is really, it's not much expensive also. So this is very precise one, but there are some, like all the technologies, there are some limitations out there. So we need to understand that the other side of this kind of technologies. Okay, and then I think I have
Also, sir, there's another question for you.
okay.
Is it neuropsychology bring ultimate solution for mental disorders rather than therapeutic approaches, as there is a corresponding neural mechanism behind every psychological phenomena?
I didn't get you. Could you please once more?
In your opinion, is it neuropsychology bring ultimate solution for mental disorders rather than therapeutic approaches, as there is a corresponding neural mechanism behind every psychological phenomena?
Oh, it's a long question, big question actually. But not only neuropsychology, because this is because when you talk from the point of the therapeutic, this is you cannot, maybe it extend to some extent this will help, but the problem is better too, because before you start the therapeutic or before the etiology, if you try to find out the etiology of this mental disease. For example, nowadays that in India also, because in our community, in Asian population, these depression or anxiety, all these are very common nowadays, because this is because the lack of the interaction, or and also the and you don't have much you don't have you don't have an enriched environment. And this is also a reason that because people will won't interact much. Okay, and that is actually even at the early stage of development also, the interaction is very less in our community nowadays. When you, because you know that when especially when you go or look around, that you will see everybody is with, even in the bus stops or in hospitals or in theater, everybody is with a small gadget, and they are very focused to their own world. So we don't know exactly what happens after the decade, what happens to, what kind of modification will happens to our system or our sensory system, but we don't know now what, because maybe that will also affect our that also increase our mental stress, or maybe that also may affect different sense organs, but we don't know yet, but it will take some time to come up with. So neuropsychology we cannot say that neuropsychology is the ultimate thing, that an approach that we can address these things, because a lot of other things are there. So everything starts from the community, from the environment, from your food, and everything is there. So there are a lot of environmental, like chemicals are there, that can also, what you will do with these chemicals. So you should start everything from your community or from society. Okay.
So another question, will cellular replacement therapy works in neurodegeneration?
To some extent it happens, some extent, not all, because a lot of the studies are going on. They also, but the problem is that you cannot, because in cellular replacement, the problem is that in neuron, the adult neurogenesis, that is very restricted in humans. This neurogenesis happens only few regions of the brain, especially in the olfactory region, and also in the dentate gyrus of the hippocampus. Only these two regions that also undergo neurodegeneration. This one neurogenesis happens even in adult. So we cannot completely we cannot say completely, we can go to some extent that cellular replacement works, but once you lose your connection, then it's really difficult to replace. That's a major problem with the neurons. So we don't know where exactly the neurons go. For example, in most of the neurodevelopmental diseases, that in the early stage, like in autism or in some other diseases, also you will see the axonal guidance. This is also a cued migration of the neuronal axons. So once they even in the early development, they lose this axonal guidance, this the synapse formation will be reduced. So that also affect, or that also leads to developmental disease. So we cannot replace that kind of, once it's formed or once the synapses are formed, you cannot replace that synapse. That's a problem. So to some extent, like therapy, cellular replacement therapy works like in case of stroke and this kind of conditions we can do, but in no, it's not applicable to all neurodegenerative diseases. Okay.
So the next question, can we expand cortical volume with stimulation?
This is not, this is this we can also, sure, definitely we can also expand the cortical volume to some extent. This is the way that usually, this is the cortical expansion is a we can say it's a simply we can say the other form of memory formation. So during memory formation, it happens in our cortical, different cortical, like in language development in the frontal lobe, or in the sensory or on sensory cortexes, or in auditory cortexes, or in visual cortex. So when you see, or when you hear, or when you sense, or everything. So all these because you will have a visual memory, you have an auditory memory, you have an olfactory memory. So everything is there. All these things are formed as a memory, a special kind of memory, and are represented in corresponding cortices. Okay, and then this has happened, that this is volume also. So this is the case when you compare with the different, for example, the evolution of brain. When you go back to the evolution of brain, you will see the from fish to vertebrate, from fish to humans, you will see the nature of the cortex, because the neocortex, the hippocampus is actually a part of the neocortex. So this is cortical volume also expand. This is expanded during evolution through stimulation. Actually, this stimulation means the information from the environment that basically stimulated the brain. And then we also know that we have a lot of foldings and everything in the brain. So of course that we can stimulate. This is the natural way of expanding our cortex, of stimulation from the environment. That's sure that it's possible, but not the we have experimentally, we have limitations to expand. Okay.
So next one, can you speak a bit about the physical mechanism behind placebo effect?
Physical mechanism that we cannot, I cannot say that completely, because we are completely a different area of research. So I cannot completely mechanism to say, I'm sorry to say in this case, because we are completely focusing in a different target. We basically study single cell target. So we are basically looking at a specific kind of cell. So in this case, the placebo effect, I cannot say completely, it's out of my box. Okay.
Okay, so next one, have you noticed any change in impulsive behavior pattern among current young generation? If yes, is food plays a role in that? Can you suggest some brain healthy food?
Brain healthy food, that means suggestion. I mean, this is one thing, is that we can say we need to be we need to have a very good enriched environment. Enriched environment means here that you will have everything because you get all the information from through your interaction, like the brain or like the neurons that interact, because the neuron, like the Hebbian postulate, the neurons, they develop or they store the memory through their reciprocal interaction. Like that, when you interact with the people, okay, when you interact with the people, then you will have a mental or cognitive enrichment, and then you read it. Then that's a one way of exercise, we can say mental exercise that you can do. And in the case of food, that I cannot say food this, but in generally, we also know that a lot of environmental chemicals are there. So we cannot, but it's better always to have vegetables. that's also boost your brain. That's sure actually. Vegetables, okay. And then you have to be not a vegetarian, but you will get a lot of either proteins from the meat also, so that will be balanced. But it's better always to have some veg. But we also at the moment we also know the situation or the condition of the vegetables, that it means a lot of pesticides are using. Even without eating, without having these vegetables you can survive. But nowadays that you will get a lot of, like the rotenone. This was actually like accidental discovery, and nowadays this rotenone was used as a pesticide before, but now this rotenone is used as a chemical, experimentally induced, it's a chemical used to induce Parkinson's in laboratories. So see the difference. So like that, I cannot say completely like that. Sorry, I'm sorry.
Okay, so next question. How social isolation and digital addiction affects cortical developments?
Okay, that's a good question. Actually, this is the cortical isolation that we actually focus. We are focusing now because we also know this autism disorders, and people will have this kind of social isolation or a kind of withdrawal symptoms, and then their interaction, their inflexibility in the social interaction. These people show social. Definitely, you will get depression. You will be depressed, and also the digital addiction, that also definitely that will in future you will also develop some kind of attention deficit deficiency, and through this kind of in future you will develop that. But and also you can, even you when you use the fMRIs, you can see the what are the areas of, for example when you image the brain at the early stage, or you can when you compare with the controls, okay, you can see what are the you using the fMRI. You can also easily identify what happens to the cortices or different other regions of the brain. For example, there was a study that is long back in 2000, I think. They studied the hippocampus of British taxi drivers, or the drivers of London city, and they found that these, because these most of these people are especially from especially they are migrants, and they used to, they know each and every corners of the London street, all the taxi drivers. And they, through these fMRI studies, they found there one of their left hippocampus is completely it's enlarged, than their volume is bigger than the normals. So they do a lot of exploratory work exercise during their lifespan. So like that, when you if you don't use your not completely, if you don't exercise your brain, then automatically there will be a chance of shrinking, the shrinkage. That is sure, that won't develop much, that will stay as such. Okay, that will definitely affect the future mental health of our society. Okay.
Sir, next question. What is the scope or role neuropsychology and cognitive psychology in field of artificial intelligence?
That's a good question. Actually, this is the time exactly that you should people, the neuropsychology, especially the psychologists, should learn the artificial intelligence. Okay, because you every day when someone come to your clinic, you will observe people, a lot of people, and okay, this is the normal way of you have a lot of observation, behavioral observe. But when you use artificial intelligence, that you can really, you can identify, you will get a lot of data from people, from the population, and you will also get a kind of prediction how they, or the to which direction the population's health or mental health is going, or which direction it goes. So this is very the artificial intelligence is applicable not only in psychology, in each and every field of biological science.
Okay, so next question. Can you explain the role 9 of prefrontal hippocampus connectivity in anxiety, depression, etc.
We can see they prefer the hippocampus, though, and so even in anxiety, depression. So the hippocampus is actually a region of the neocortex that they will get all the information from different sensory modalities, and then when it reaches to the hippocampus, and that also modulates its activity, and then if it is necessary or of the information is relevant for the brain, that will try to incorporate or try to consolidate it. But we cannot simply say it's a single region is responsible for this anxiety or depression. It's also because the principal transmission of the brain, or the neurotransmitter of the brain is the glutamate and also the GABA. So wherever when you see a kind of imbalance of this synaptic or the excitatory or inhibitory transmission, then definitely you will see you will develop a kind of anxiety and depression. So that we cannot pinpoint a single point is responsible for this kind of behavior or the anxiety or depression. So this is basically a network. So this network contribute basically the output of the behavior. So we cannot simply pinpoint this is the region it's responsible for. So especially in the case of this anxiety and depression, because these are more related to the glutamate and GABAergic transmission. So we cannot simply pinpoint. So wherever it happens, wherever the transcription or the GABA and glutamate is there, then you will have a chances of this anxiety and depression. Okay.
Sir, can you explain a little bit about activity-dependent plasticity?
So activity-dependent plasticity, basically, this is the region that is basically in the CA1. I explained in the slides. The CA1, the hippocampus. And when you look at very closely in the hippocampus, you will see two types of cells are present. One is the pyramidal cell layer, and also the second is dendritic granular cells are present in there. So the CA and the pyramidal cell layer that forms like a small horn-like structure, and that is called it is also because its name is the CA name is Cornu Ammon's horn, because of its resemblance with the Egyptian god Ammon's. So the Ammons has a horn like this, and the structure basically represent the Ammon's horn. So that is the reason it got the name the Seer region, Cornu Ammon's region. So and in the Seer region, you will see there are subdivisions are there. Then CA1, CA2, and CA3 regions are there. In CA1, this is the region where the activity-dependent plasticity is there. Activity-dependent plasticity mainly responsible. This is achieved through the Nmda receptor-dependent plasticity. So the Nmda receptor is basically responsible for the activity-dependent plasticity, because when the incoming or that these neurons that or these channels works depends on the frequency of the incoming information or the incoming frequency or incoming information is that much strong, then only this channel will open. On that way, then it starts its mechanisms to then it identify this information is relevant for the system and to be stored in different regions of the brain. That is the way it happens in the CA1 region. That is also I did my PhD and everything in the activity dependent plasticity of the hippocampus. Okay.
Sir, how agenesis in cortical structures affects behavioral realm?
This is how the agenesis. This is basically very relevant thing that you will see especially in the case of you have seen these slides of the Alzheimer's disease. The cortex will also during the age So then you will have a kind of saturation, because you are a lot of process will happen, things will happen, because in age then you will have the reduced receptors, and also the responsiveness is also reduced, and the cortical regions will also shrink, they will also shrink. So a lot of things will happen in the neurons also, and also the neurons have a chance of getting different plaques formation. So all these also affect our behaviors. So so for example, in different we also know that different behavior that is expressed uh, in aged people, and these are basically due to these shrinkage of cortex, or maybe some the saturation of the activity, and so a lot of things are there.
That's it, sir. Thank you so much.
Okay, thank you very much. Thanks a lot.
Over to you, Vinya.
Okay, thank you.
Thank you, Shimna. Thank you so much, sir, for clearing all our doubts and for spending your valuable time with us. Now, let me invite Ginya, second year basic psychology student of LISSAH College, to propose the word of thanks. Over to you, Ginya.
Hello, am I audible?
Yes, ma'am.
A warm and graceful morning to one and all. It's my privilege to propose vote of thanks and acknowledge the contribution of those who worked really hard to make this happen. I, Ginya, on behalf of LISSAH College and the entire fraternity of the institution, first of all, extend my most sincere thanks to the Almighty God for making today's event a resounding success. On behalf of my college, I extend a really hearty vote of thanks to the chief guest of the day, Dr. Binu R, who spent his busy schedule to grace this occasion. Today, we had an opportunity to hear you, sir. Thank you so much. Next, I would like to thank our management and head of the department for giving us this opportunity. Special thanks to the coordinators led by Fida for their efforts and support to help organize this webinar. I would also like to thank the backbone of our team, our technical team, for their hard work. Last but not the least, I would like to thank each and every one of you for attending this webinar and supporting us. Once again, thank you all for being with us. Have a wonderful day ahead. Thank you.
Thank you very much. And also, once again, I give my great pleasure and also big thanks to Dr. Emerson for inviting me for this talk. And also it was really an interesting interactive session. And also I thanks all of you for this talk, and thanks once again to the management and also all the faculty members and students of the LISSAH College. Okay, thank you very much.
Thank you, sir. Thank you, Ginya. The feedback form is now put in the chat box and comment box of Zoom and YouTube respectively. Please do take time to fill the form and let us know your valuable feedback. Here we come to the end of the program. Thank you everyone for your support and cooperation. We'll see you next time. Until then, thank you and have a nice day. You can leave the meeting now.